Assalamu alaikum. My name is Kalsumi Chas and my topic of presentation is PCR and its three steps. First of all, PCR stands for polymerase chain reaction. In simple words, polymerase chain reaction is a technique that is used to make several copies of a small fragment of DNA. If you understand the meaning of the words in polymerase chain reaction, we can better tell about this process from its name itself. How? Polymerase chain reaction. Polymerase means an enzyme that make polymerase of any other molecule, which in this technique is DNA. On the other hand, chain reaction. Chain reaction is a type of chemical reaction which progresses in an exponential way. Or in simple term, if first reaction produces two molecules, the second reaction will make four and the third will make six copies and so on. So in a matter of just a few hundred reaction, we can produce billions of copies of single fragment of DNA. PCR was developed in 1983 by Carey Mullis and it has three major steps: denaturation, annealing, extension by DNA polymerase. PCR components, we can say that things we needed to perform PCR: DNA template, primers, tag polymerase, deoxynucleotide triphosphate nucleotides, PCR buffers, and thermocycler. Commonly used equipment in PCR is the thermocycler and also called as PCR machine. Inside the PCR machine, we have these small tubes in which all the chemicals are inserted and the reaction takes place. Now look at the stuff that we put inside these tubes and makes this reaction possible. First of all, RNA template. RNA template is a segment of original DNA which we want to amplify. Primers. Primers, second important thing that we need to perform PCR are the primers. DNA polymerase need primers to start the reaction. Primers bind the template DNA by complementary base pairing. In this way, we are able to select a particular region of the DNA that we want to amplification. Tag polymerase. Tag polymerase is a type of DNA polymerase, and like DNA replication in any organism, PCR require a DNA polymerase enzyme that makes new strands of DNA using existing strands as template. The DNA polymerase typically used is PCR is called dark polymerase after the heat tolerant bacteria from which was isolated called Thermus aqueous and naturally lives in hot spring and the most active around 70 degrees Celsius. The heat stability is ideal as high as needed for the reaction. Nucleotides. As we all know that nucleotides are basic building blocks used for DNA synthesis. PCR buffer also added to the PCR tube and thermal cycler is known as PCR machine. Next is step 1, denaturation. Denaturation in which PCR, we rise the temperature of the machine to 94 to 98 degrees Celsius. This is done to denature or separate the two strands of DNA. As a result of this step, we got two separate strands of DNA and the breakage of hydrogen bond between complementary bases. Next step is annealing. Before the getting in detail, do you know the meaning of annealing? Basically, this word was used in metal industry where they heat a certain metal and increase its temperature and then cool it. This process is called annealing, which is done to remove the internal defects of the metal. Same in the case in the PCR. In PCR, first step, we increase the temperature and in annealing process, we decrease the temperature. We lower the temperature 50 to 56 degrees Celsius. And primers are usually 20 nucleotides base pair long sequences complementary to template sequence. Primers bind to their complementary sequences and primers in the PCR tube can bind to the target sequence on the single stranded DNA. Next step is extension. In extension, we further increase the temperature which is 72 degrees Celsius, so that the tag polymerase extends the primer synthesis new strands of DNA. The tag polymerase adding new nucleotides to the short sequences of the primers to form new strands of DNA which complementary to the original strands. We can got two double DNA. One is the original parent DNA and the other one is the new DNA formed by the tag polymerase. DNA polymerase bind to the annealed primers and extend DNA at the three end of the chain. Next is exponential amplification. Due to the exponential nature of the chain reaction, we are able to use billions of copies of DNA fragments from only a single copy of fragment. The product of first reaction used as a substrate for the next reaction. Next is applications of PCR. There are many uses of PCR, such as in the field of medicine, genetic researches, evolution study, PCR in research, diagnosis, etc. and diagnosis infections. For example, it is used to diagnose infection by virus, as we take blood sample of the infected person and then infected virus, then we can amplify the DNA of the virus so that we can able to study which type of virus it is and its all properties. In conclusion, it is introduced in 1980s and PCR continues to prove to be a useful tool with broad application in discovery biology, medical diagnosis and agriculture. This is all from my side. You have any question you can ask. Thank you so much.